My experiment was a response to a previous experiment done by a group of researchers attempting to create a device that would rapidly detect Staphylococcus aureus. They attempted to create this device by immobilizing antibodies on a gold surface, and by running electric current through two of these gold surfaces while running liquids over them. If there was presence of bacteria Staphylococcus aureus in the liquid, then they would be caught by the antibodies and then stuck to the surface along with the immobilized antibodies. And if this occurred, then there'd be a small but noticeable impedance in the electric circuit. This could be registered by any machine. Therefore, one could detect the presence of Staphylococcus aureus by a simple little impedance. Such a device would be incredibly useful in the field of detection for something as dangerous as an antibiotic-resistant bacteria, which is what these researchers were told to create. They created the entire device from scratch, essentially. And according to the results I was received, they were not successful in creating a functional device. So thus, the project was brought on to me. And since I'm a chemist rather than an engineer, I would double down on trying to find out the mechanism behind how it worked. And according to all that I was given about the previous experiment, the people behind it did not know a thing about the chemistry, the chemical mechanics in the device. They didn't really know how the antibodies immobilized. They were simply drawing on previous research. Previous experiment did the exact same thing. So their cited resources was just a chain of these experiments doing the same thing, with little bits, but none of them really discussing the chemistry behind it. So I performed a great amount of research trying to find out how this works or how it should work, because to my knowledge, organic compounds shouldn't stick to metallic gold, not unless it's some sort of adhesive, but at that point it's like a micro suction cup or something. Or like things are just held together by like suction forces, molecular level. But what they were discussing was these antibodies were held on by bonds to the metallic gold. And that really baffled me, because to my understanding of chemistry, metals really only bond to non-metals if there's an ionic state of some sort. However, after quite a bit of research, I found that the molecules that are binding to these antibodies first, which then bound them to the gold, has sulfur substituent. And it was from that led me to learn that there's a unique property between gold and sulfur that makes this whole thing work. But sulfur substituents can form unique bonds to metallic gold, and these should be strong enough to hold the antibodies in place. So therefore, the chemistry was to bind these antibodies with these molecules that had sulfur substituents, and then thus the sulfur substituents would bind it to the gold and mobilize the antibodies on the surface, thus making the whole thing work. And according to the procedures I received through the previous experiment, scientists or engineers used, they basically washed off their necessary materials before they actually even bound them to the antibodies. They washed off the molecules that had the sulfur before they were connected to the antibodies. And then some, they had too many rinses, and the reaction doesn't carry out. So I doubled down on the reaction, but I kept the concentrations of the antibodies the same, so that I can at least see what levels of antibodies they are working from. This had some difficulties because they recorded their ratios of antibodies to solution as ratios, but also in volume. So I'm not even sure if I had the exact same concentration as they did, because they utilized volume instead of, well, molar concentration. So I had to assume while I was working with the same stock of antibodies concentration they were, that I diluted to the appropriate ratios, or best I could. I recorded all the concentrations in moles this time, so it'd be easier for other people to understand in the future, but I don't think they were too fixated on the chemistry of how it worked, rather they were more centric on the whole device. Whereas my experiment was just trying to determine that the antibodies can be stuck to the gold surface. I think that helped my focus my research a bit better than theirs. So what I did was I had four different solutions with one gold tile each, and each gold tile at the end would be analyzed with XPS or X-ray photoelectron spectroscopy, and from there I would then compare the results of each tile to try and see if there actually was antibodies present on the gold surface. Two of the gold tiles had the different concentrations of antibodies in the solution, though the highest and the lowest from the previous experiment, while the other two are two levels of control. First, containing just the solution that all this was done in, a phosphor buffer solution, PDS, and the next one is the phosphor buffer solution plus the two chemicals used to bind the sulfur substituent to the antibodies. So if there's no antibodies present, will we still get a noticeable signal? That's kind of the goal. As of this moment, I have not yet received back the results of the analysis, so the video has been a bit delayed, but it's coming out now. But I do not have the results of whether or not they found anything on the gold surface. However, I would still like to discuss what I've done so far. As I said, I was doubling down on the mechanics behind the chemistry of it, since my field is chemistry, and it's clear that they didn't really understand much about it. And it took quite a bit of research to find out is this sulfur-gold interaction. Whether or not the antibodies will be functional in this state, being bonded to these other molecules and stuck on the gold surface, I'm not sure. However, theoretically, they should be immobilized on the surface, so that's a start. Whether or not they detected XPS is also up for debate, but I'm quite confident that if it all went well, which I felt very cleanly as possible in my lab, and everything else, they should find something at least. Though I will say the concentrations of the antibodies were much too low for real significant results you need to use the device. So in the future, if I were to continue this experiment, I would preferably like to move to higher concentration of antibodies for functional use in this kind of project, as well as more trials and experiments, because this kind of testing this should be done with many samples to ensure that this size and small scale should be properly analyzed. And on top of all that, I would also like to check other conditions such as temperature, rate of adherence to the surface, such as how fast is the rate of bonding for the sulfur to the metallic gold, and such things as that. As well as I like to test how sturdy it is when being transported or being formed, because the way I process it and the way the other people process it, they simply place them in solution. Well, according to the past procedure, the past people doing this, they simply poured the solution over the gold surface and having the antibodies bond while the fluid is running. That wouldn't be very stable. That wouldn't be very enough time for them to form stable bonds, even if they were functionally present. However, I simply left them in solution in closed containers, as well as I try to keep all my containers enclosed at all times, as well as try to keep them refrigerated, because I know antibodies themselves tend to break down at room temperature. So, if anything else, I would like to continue this research if I was given the opportunity to test different variables like time, temperature, even exposure, because nothing is quite perfect when transferring the solution from container to container. as well as I suppose that's it. I wish I could be in person for this oral presentation, but due to COVID, I can't really proceed to the questions section. So I guess if this is put onto YouTube, then I suppose leave something in the comments below. This has been my senior project for 2020, and I hope I impressed you somewhat with potential ideas for the future of medical diagnostics, even if my role is just the basics of can this work.